the centrifuge tube with the sample is going to undergo decentrifugation, a centrifugal field is going to be applied where there is a fixed centrifugal field. There is no changes in the centrifugal field. After the centrifugation, a sample components based on its density, they are going to be separated. A lesser density compound is going to be separated at the top, and high density compound is going to be separated off at the bottom. So from the centrifuge tube, a different density components or a different gradient, so it must be separated without disturbing of other gradient separated components. So the recovery is very important in this centrifugation and very tedious process. So for this, a recovery of a sample can be done by two methods. One is puncturing technique and other is displacement technique. In the case of puncturing technique, where that is, from the name we can found that a puncturing technique involves a puncturing of test tube. So the test tube is going to be punctured off with the needle at the bottom, where it is going to have a higher density component separated, and at the top where there is a low density components is going to be there. By without disturbing the low density component, high density component is going to be separated off by puncturing. A high density component is going to be separated off in the fraction test tubes. After the high density components is going to be collected off with the fraction test tubes, then another test tube can be used for low density component collection. So a dropping medium which is going to consist of components which is of its similar cell components or biomolecular components. A peristaltic pump can also be used for sucking of the medium from the hole. It can also be clogged, so clogging can be possible, which is going to be not possible if a peristaltic pump is used. Next, a displacement technique. In the case of displacement technique, from the name itself we can identify that each cell component with its gradient is going to be displaced off by this technique. From the puncturing technique, we have does not disturb any other component, where the component is going to be separated by puncturing at the test tube bottom and separated based on manually with the tedious process. But in the case of displacement technique, where there is a cover at the outlet,